This is an A-level biology presentation looking at in vivo DNA technology. In this session, we're going to cover what is meant by the term recombinant DNA technology, and we're going to look at methods involved in isolation of genes to produce DNA fragments, and we'll bring in the concept of sticky enzyme restriction endonucleases. To get started, describe the process of transcription and translation just using keywords. You're not looking for sentences, you're only looking for keywords. Remember, these are the words that are going to appear in the mark scheme. Make sure you go back over transcription and translation and the other presentations should you need to. Pause now, come up with as many terminologies as you can. Remember, in transcription and translation, we are producing an RNA strand from a DNA strand in transcription, and we are producing polypeptides or proteins in the process of translation from messenger RNA using a number of other things. If we start going into it more detail, you need to remember start referring to the template strand, the enzymes helicase, hydrogen bond, unzipping. These are all the terminologies you should be using in transcription. And in translation, you should be using the terms codon and anticodon. You should be referring to a large and small subunit of the ribosome. You should be talking about triplet codes, polypeptide bonds between amino acids. You should talk about P site, A site, transfer RNA, and you need to make sure that you can talk about those in sequence. In terms of DNA technology, the genetic code adenine, thymine, guanine, cytosine, and uracil is universal. Every organism on this planet is based on that concept. When we have transcription and translation, these are processes that are also universal. They are common to all organisms regardless of where we find them in terms of everything from little viruses all the way up to the most complex multicellular organism. What that means is that we can actually start to take DNA from certain organisms and start to move it and give it to other organisms. And that's the concept behind transgenic organisms and genetic engineering. Well, we need to start off, what is gene cloning and why is it important? Well, pause for a second and think of what do you think is meant by the term gene cloning and why do you think it's important? After a minute, restart the presentation. Pause now. Well, gene cloning is the ability to isolate and replicate a specific gene of any organism. We are not cloning the entire organism, we are just cloning one particular gene. It's important because it allows us to produce large numbers of polypeptides that are coded for by that gene using microorganisms. Now, when we talk about in vivo gene cloning or gene technology, we're actually talking about a number of stages of a process. We start with the isolation part, we then do an insertion, transformation, identification, and growth and cloning. And those 5 stages combined make up in vivo gene cloning. To start with, we're going to look at gene isolation. So how do we actually isolate the gene we want? And there are 3 ways we can isolate the gene. We can isolate the gene working with the DNA directly. We can isolate the gene from messenger RNA and use that. Or we can isolate the gene using a protein sequence in a gene machine. And we're going to look at each of those different mechanisms in more detail. Remember, the sole purpose is to end up with the genetic code that represents a particular polypeptide, a particular gene. Well, to start with, if we have a strand of DNA, what we can use is a group of enzymes called restriction endonucleases. And the restriction endonucleases are able to cut DNA double helixes at specific sites. And when they cut them in a staggered formation called a sticky end. So rather than just a straight chop in the middle like you can see in the yellow example at the bottom with the blunt ends, we have this overhang. And the reason we find that overhang so useful is because when it comes to inserting that particular gene back into another host organism, we can use that overhang to make sure the gene's inserted in the right place. If we have blunt ends, it's a lot more difficult to actually put that in. The second thing we can do is we can start with the messenger RNA. And if we start with the messenger RNA, we can basically reverse engineer it to form the DNA double helix. And how we do that is, first off, we extract the messenger RNA. Remember, the messenger RNA is extracted from the cytoplasm, not the nucleus. When you have a messenger RNA, you have a gene. The natural process of transcription has already got a polypeptide code, and it has turned it into messenger RNA sequence and removed any introns that are there. So we've only got the coding region. The next thing we do is we add an enzyme called reverse transcriptase. And reverse transcriptase is able to build DNA strands from complementary RNA strands. Once we have the complementary DNA strand, shortened to cDNA, we then need to turn it into a double-stranded DNA molecule. So we have to first break up the messenger RNA that we used as our template originally, and we do that using what's called hydrolyzing enzyme. Once we've got the hydrolyzing enzyme to remove the messenger RNA, we're left with a cDNA strand. As with all DNA, if we want to make a complementary DNA strand to a DNA strand, we use the enzyme DNA polymerase. And the DNA polymerase reads the cDNA and forms a complementary strand. And what we end up with is a double-stranded DNA that contains the coding sequence that is complementary to the original messenger RNA that we took out of the particular host cell. Now, the third way we can get that DNA sequence is we can use what's called a gene machine. And a gene machine is literally a computer that you punch in the sequence of amino acids and it builds a particular code for you. We then use a process called PCR, which we'll come into in a later presentation. And what PCR does is PCR just makes large numbers of copies of any particular DNA strand. And then, of course, what we'll do is we will get that DNA strand and we'll insert it into a bacteria plasmid, and that bacteria will then start to do the transcription and translation process and produce the polypeptides linked to the particular gene that we have produced. Be aware, we do have this term oligonucleotides, and oligonucleotides are where we get short sequences of overlapping DNA. Rather than the gene machine producing one big giant DNA sequence in one go, it produces lots of little overlapping sections which then fit together a bit like a jigsaw. So to finish off, we've got a little table. What you need to do is, for each of the three methods of isolating a DNA strand, restriction endonucleases, reverse transcriptase, or gene machine, you've got to identify what the source material is and also identify if introns are going to cause problems with that particular method. Pause the presentation now to give yourself time to work it out, and when you're ready you can restart. So in terms of source material for restriction endonucleases, well, restriction endonucleases work on DNA. Reverse transcriptase, well, with reverse transcriptase, we will take only the cell that makes the required polypeptide. Restriction endonucleases will work on any cell because all cells in an organism have exactly the same DNA. However, when we specialize cells and cells become differentiated, they only produce certain messenger RNAs depending on the function of that particular cell. Hence, the reverse transcriptase will only work with a cell that's actively producing the messenger RNA that we are targeting. In terms of a gene machine, Well, the gene machine, again, we need to know the polypeptide that is going to be extracted. We need to know the primary structure, the sequence of amino acids. So again, we need to go to the cell that is actively doing transcription and translation of that particular bit of DNA to produce the polypeptide. And then we work out the amino acid sequence. And then from there, we can obviously go back all the way to work out the original DNA sequencing. In terms of introns, restriction endonucleases, well, they are not intron free. Because we're going to the source DNA, there are going to be introns there, and that can obviously cause complications. In reverse transcriptase, we are intron free. There are no introns involved because they have been taken out in the process of splicing. And when it comes to gene machines, again, introns are not a problem because we are only targeting the coding regions. Remember, if we're dealing with a prokaryotic organism, then we do not have any issue with introns at all. There's a couple of other features just to be aware of. Remember that restriction endonucleases are specific to certain regions of DNA, which is why they will cut at certain points. And the gene machine is, of course, fast and accurate. It can produce any sequence you want. But we do have this thing about the degenerate code and the fact that even though we have the right sequence of amino acids, because each amino acid can have more than one DNA sequence coding for it, we may not actually have the exact same DNA sequence as the original organism. However, we do have a sequence that will produce the polypeptide we want.